Hello everyone, welcome to my YouTube channel. Today's topic is the cell and its important organelles. So let's start. Firstly, what is the cell? Cell is the basic structural and functional unit of body or life. So what is about the cells? Have you imagined how many cells in a body of animal or human? Maybe their number is in the trillions. The number of cells is estimated to be about 100 trillion in the human body. Each of these cells had their start beginning with fertilization of an oocyte. What is this? This means that each and every cell of the body have their origin from which? From a single cell, the zygote. When the zygote is formed, then the mitosis, the cell division occurs, and the cells of the body are formed. So, the origin, the start of each and every cell in the body begins with the fertilization of an oocyte. The appearance of cell varies with the organ in which they are part and will be shown and described when encountered. Cells are highly organized chemical systems. Also, the cells, the appearance of cells, like they vary in the size, shape, function. In a similar pattern, the appearance of cell varies. Different tissues in the body, different organs in the body have different characteristic of cells. They are specialized. Like nervous system have neurons, blood cells have RBCs. Different type of cells in the body have different appearance. Also, cells are highly organized chemical systems. It is correct. Cells are highly organized because if there is no organization in the cell can't be the functional unit. Okay. Cells also digest, respire, excrete in the same pattern as the animal body or human body does. Now, the structure of the cell. The basic component of a cell are the plasma membrane or cell membrane that bound the cell and give it limits. जो बेसिक कंपोनेंट्स में है वो हमारा प्लाज्मा मेम्ब्रेन होता है, also known as cell membrane, and the next is cytoplasm, and the next is nucleus. As we know, the three basic components of a cell: plasma membrane, cytoplasm, and nucleus. What is plasma membrane? Plasma membrane bound the cell and defined its boundary, while cytoplasm is the homogeneous ground substance in which the formed elements are suspended. Means the cytoplasm is the ground substance in which the organelles that, or the granules or substances are dissolved or suspended. And the third is nucleus, control center of the cell. The nucleus is separated from the cytoplasm by a nuclear membrane, and the cytoplasm is separated from the surrounding fluid, interstitial fluid, by a cell membrane. Also, there is separation. There is a boundary of each and every component. Like nucleus is separated from the cytoplasm by nuclear membrane, while cell is separated from the surrounding fluid, that is known as interstitial fluid in the body, by cell membrane. The cell membrane is usually pliable and composed of phospholipid and proteins. The most important line: the cell membrane is usually pliable, जनरली सेल मेम्ब्रेन लचीला होता है, pliable मीन्स लचीला, and composed of phospholipids and proteins. Phospholipid and proteins का बना होता है सेल मेम्ब्रेन, the most important thing. The phospholipid molecule occur in two layers. What about the phospholipid layer occurs in two layer. Also the protein molecules associated with them, they are also true. And the protein molecule associated it may be on outer layer or inner layer and may penetrate completely or incompletely. Because of cell specialization, no cell can be called typical. The cytoplasm is the location of metabolic activities and is filled with both minute and large dispersed particles and organelles. Sorry. देखिए एक लाइन लिखा है कि बिकॉज ऑफ सेल स्पेशलाइजेशन नो सेल कैन बी कॉल्ड typical. What is typical cell? A typical cell is the representative of all the cell. That is, they have basic structure of plasma membrane, cytoplasm, nucleus. But do all the cells in the body have all these structures? No. Like mature RBCs, they are divided of the nucleus. So no cell in the body can be called as typical because they are specialized. Because of the specialization, no cell in the body can be called as typical. The cytoplasm is a location of diverse metabolic activities. Yes, metabolic activity is very important for the survival of the cell and the living organism. It occurs in the cytoplasm and is filled with minute and large dispersed particles and organelles. Already discussed, filled with both minute and large dispersed particles and organelles. Next is the organelles, the most important component of the cell. What about organelles? First, we see the diagram of a cell. How the cell look like? How the animal cell look like? This is the figure of a animal cell. See, in the center, the nuclear envelope which separates the cytoplasm, the nucleolus which is found in the nucleus, smooth endoplasmic reticulum, rough endoplasmic reticulum, mitochondrion, nuclear pore, lipid inclusion, secretory vesicle, Golgi complex, centrosome. All these structures are found in the cell. So we will study one by one. Let's start. Firstly, we know what is organelles. These are highly organized physical structures. These are highly organized physical structures present in the cell. In addition to the cell membrane consists of the nucleus, endoplasmic reticulum. I will tell you all these structures. The organelles name. If you don't know about the organelles, just read nucleus, endoplasmic reticulum, Golgi apparatus, mitochondria, lysosomes, centrioles. These are the examples of the organelles in animal cell. These structures assist the cytoplasm with its metabolic activity by receiving maternal material into the cell, synthesizing new substance, generating energy, packaging material for transport to other parts of the cell or to circulate excretion of waste product and reproduction. So you can see sorry a diverse function of the cells these organelles performing, like firstly diverse metabolic activities occurring in the cytoplasm, as you know. The next is receiving materials into the cell. the materials received by the cell, synthesis of new substance, generation of energy or generating energy, packaging material for transport to other part of the cell or to the circulation, excretion of waste, reproduction. All these functions are performed by the individual cell, just like a whole body. I will already tells you. Okay, the first organelle is the nucleus. What about nucleus? See, the nucleus is the control center of the cell, controlling its chemical reaction and reproduction. Nucleus is the control center of the cell. What is controlling? Reproduction and chemical reactions. It is controlling the chemical reactions and the reproduction. Biochemistry or biochemicals means sorry means the chemical reactions occurring in the living body is the biochemistry or biochemical. So these biochemical reactions are controlled by the nucleus and reproduction also. It contains large quantities of DNA. Obviously, DNA is found in the nucleus. Nuclear component consists of a nuclear membrane, one or more nucleoli, and chromatin, all bathed in nuclear sap, that is nucleoplasm. As we know, nucleus. But in the nucleus, there are also structures found in the nucleus, and these are nuclear membrane, nucleoli, and chromatin. And all these are bathed in nuclear sap or nucleoplasm. The nuclear membrane, also called nuclear envelope, consists of two membranes, wherein the outer membrane is continuous with the endoplasmic reticulum, and the space between two nuclear membranes, the lumen, is also continuous with the endoplasmic reticulum. The nuclear membrane, which separates the cytoplasm and nucleoplasm, is very. See, what is this? This is continuous with the endoplasmic reticulum. The outer membrane is continuous with the endoplasmic reticulum, and also the space formation between the two membranes of the nuclear envelope. This is also continuous. The space, the lumen, is also continuous with the endoplasmic reticulum. Nuclear pore penetrate both layers. The nuclear pores present on the nuclear envelope penetrate the both layers because these nuclear pores act as the path for the transport of materials between the cytoplasm and nucleoplasm. The, the pores permit exchange between the nucleoplasm and the nucleus and cytoplasm of the cell. Already told, including movement of RNA synthesized in the nucleus. The example is movement of RNA synthesized in the nucleus out into the cytoplasm. Obviously, as you already know, the gene expression, all the information coded in the DNA. DNA से RNA बनेगा, RNA से proteins बनते हैं. These RNA must be come out to the cytoplasm for their expression, complete expression, the synthesis of specific proteins. The nucleolus does not have a limiting membrane and is a structure that contains large amount of RNA. Okay, the nucleus does not have a membrane like nucleus. Okay, and it is contains RNA and proteins. Means the nucleolus is made up of RNA and proteins that are found in ribosomes, similar to ribosomes. The next is chromatin. What is chromatin? Chromatin is appears as dark staining particle throughout the nucleoplasm in the non-dividing cell. Very important thing. In a normal cell, it has two phase dividing phase and resting phase. The cell which is in dividing phase have chromatin organized into chromosomes. You will study in cell division chapter about the chromosomes. Okay, the next is endoplasmic reticulum. Sorry, the endoplasmic reticulum is a network of tubular and flat vesicular small thin walled cavity structure in the cytoplasm that all interconnect with one another. As already discussed, the endoplasmic reticulum is continuous with the outer layer of the nuclear envelope. Okay, so what is this? This is a network of tubular and flat vesicle structure in the cytoplasm. Tubular and flat vesicles, just like cavity, small thin walled cavity, have vesicles. These are cavity. Okay? These are structures present in the cytoplasm, and these structures are interconnected with one another. Interconnected, so the fluid within the lumen of the endoplasmic reticulum is continuous with the fluid in the nuclear envelope. Already told, the space is continuous with the endoplasmic reticulum space. A similar pattern, the fluid in the lumen of ER is continuous with the fluid in the nuclear envelope and is different from the fluid in the cytoplasm. The fluid present in the cytoplasm is different from the fluid present inside the endoplasmic reticulum. A large number of small granular particles called ribosomes are attached to the outer surface of many parts of the ER. Small granular particles. These are ribosomes attached to the outer surface of the endoplasmic reticulum. When these are present, they are called the granular or rough ER. Where they are not present, they are called agranular or smooth endoplasmic reticulum. Simple. There are two types of endoplasmic reticulum: smooth endoplasmic reticulum and rough endoplasmic reticulum. Where the small granules are present is the granular or rough endoplasmic reticulum. Where these granules are not present, this is agranular or smooth endoplasmic reticulum. Okay. What is the ribosomes? These ribosomes is composed of a mixture of RNA and proteins. These are composed of RNA and proteins. And what is the function of ribosomes? Ribosomes function in the synthesis of protein. प्रोटीन्स का सिंथेसिस करते हैं. The agranular endoplasmic reticulum functions in the synthesis of lipid substance and other enzymatic processes of the cell. The agranular endoplasmic reticulum. What the function of the agranular endoplasmic reticulum is the synthesis of lipid substances. The hydrophobic lipids are hydrophobic in nature. So the synthesis of lipid substances occurs on a smooth endoplasmic reticulum. The next organelle is the Golgi apparatus. So the Golgi apparatus is closely related to the endoplasmic reticulum. It is prominent in secretory cells, being well developed in cell secreting enzymes and hormones. The most important, but where the secretions are important, like secretory cells में very prominent, काफ़ी अच्छे खासे नंबर पाए. Being well developed in secretory enzymes का secretion हुआ, hormones का secretion हुआ. In these cells, Golgi apparatus have a very important role. It packages material made in the cell and transform them into units that are then distributed outside the cell. So what is doing? It pack materials within the cell and transform them into units that are then distributed outside the cell. The packaging begins when vesicle pinch off from the ER and then fuse with Golgi. How this happens? The packaging begins when vesicles pinch off from the ER. Means the packaging start when the vesicles pinch off from the ER and fuse with Golgi. Means it is closely associated with the endoplasmic reticulum. The whole packaging mechanism is associated with the endoplasmic reticulum. The vesicular substance are then processed in the Golgi. The vesicles the vesicles come from the endoplasmic reticulum and then processed in the Golgi. Okay, the vesicular substances are processed in the Golgi and to form lysosomes and other secretory vesicles. Lysosomes, what? Lysosomes contains the digestive enzyme. If they burst inside the cell will die. Why? Because it digests the own cell. Okay, so lysosomes and other secretory vesicles which become surrounded by membrane, and lysosomes secretory vesicles, these are surrounded by membrane, unlike nucleoli. They are then released from the Golgi apparatus for storage or use in the cell or transported to the cell membrane where they release into the extracellular fluid as secretion. So very simple. They are released from the Golgi apparatus for storage or use in the cell or transported to the cell membrane, and the cell membrane release them into the extracellular fluid as secretion. Simple. The milk secretion, enzyme secretion, the hormone secretion, all the cells have good amount of collaborators which involves the packaging, processing, and then secretion. Next is mitochondria, but we already studied in previous lecture. So we continue from lysosomes. Lysosomes. The lysosomes, the vesicular organelles called lysosomes, are formed by the Golgi apparatus and then become dispersed throughout the cytoplasm. These are also vesicular organelles, already told, because they are pinch off from the endoplasmic reticulum and then processed in the Golgi apparatus. So these are vesicular organelles formed by Golgi apparatus and then become dispersed throughout the cytoplasm. Because of some contains digestive enzyme, their presence in the cytoplasm provide an intracellular digestive system, allowing digestion of damaged cellular structures, food particles ingested by the cell and bacterial cells. So what is the function of these lysosomes? It contains the digestive enzyme. They are distributed throughout the cell and these provide intracellular digestive system, allow the digestion of damaged cellular structure, food particles ingested by the cell and the bacteria. Okay, lysosome is very important. The next organelle is the centrosome. The centrosome is located in the cytoplasm close to the nucleus and contains two centrioles. Means the centrosome located in cytoplasm obviously, but there is a point what close to the nucleus and contains two centrioles. What is centrioles? The centrioles are the structures of microtubules inside the centrosome, and these centrioles are oriented at right angles to each other, and each consists of nine group of three microtubules arranged in a circle. During cell division, the centrosomes serve as spindle pole and help to organize the microtubules. So, centrosome function क्या है? These have role during the cell division, serve as the spindle pole, and organize the microtubules. They are made up of centrioles, which are at right angles to each other, and these centrioles made up of nine group of three microtubules. I hope you will understand all these organelles. The last organelle which we left is the mitochondria. So the most important organelle is the mitochondria, because if there is no energy, there is no chance of survival of the cell. So mitochondria are the powerhouses of the cell because they are the principal site for energy production. Mitochondria is also known as powerhouse of the cell because they are the site for energy production. The number in a cell depend on the amount of energy required, and mitochondria increase in number when cellular need increase. माइटोकॉन्ड्रिया का जो नंबर होता है किसी सेल के अंदर, it depends on the cellular need. Their number increase as the cellular need increase. Mitochondrion is composed of an outer and inner membrane. The I'm talking about the structure of the mitochondria. So it is made up of outer and inner membrane. The inner membrane has enfoldings that provide the cells for attachment of oxidative phosphorylation enzymes. So the inner membrane is enfolded or enfoldings that provide cells, and these cells have attachment of oxidative phosphorylation enzymes. These enzymes have a role in the production of energy, like ATP. The inner cavity consists of matrix supporting substance that contains enzymes and coenzymes required for extraction energy from nutrients. The inner cavity matrix also have enzymes and coenzymes or cofactors. What is the role? They extract energy from the nutrients. Nutrients like glucose, amino acid, free fatty acid, and other. The matrix is the site of the citric acid cycle, the very famous TCA cycle or Krebs cycle. So, the matrix of the mitochondria is the site for citric acid cycle. Very important cycle, the tricarboxylic acid cycle or the Krebs cycle. Okay, thank you. I hope you all understand these concepts of cell and its organelle.